Welcome to Department of Botany, Kalna College, and this is Somnath Mondal, and today's topic is regulation of tryptophan synthesis in E. coli. So, TRP operon controls the synthesis of enzymes taking part in tryptophan amino acid biosynthesis. Now, this is the overall pathway of tryptophan biosynthesis from chorismic acid. Now, this is a multi-step process, and each and every step we need some certain enzymes. Here comes the role of TRP operon. It has 5 structural genes, namely TRPA, TRPB, TRPC, D, and E. TRPE develops after transcription and translation eta subunit, and TRPD forms the delta subunit. Now, these two subunits combine to form anthranilate synthetase enzyme. This enzyme is responsible to catalyze the chorismic acid to convert it into phosphoribosyl anthranilate. So the first two steps of the biosynthetic process. Next, TRPC gene after transcription and translation produces indole glycerol phosphate synthetase, and indole glycerol phosphate synthetase enzyme will catalyze the conversion of phosphoribosyl anthranilate into carboxyphenylamino deoxyribose phosphate or CDRP, and finally into indole glycerol phosphate or INGP. Now, TRPB gene will produce beta subunits and TRPA the alpha subunits. Beta and alpha will combine to form tryptophan synthetase enzyme, which will convert INGP into tryptophan. Now, as in all operon, in TRP operon there will be promoter, operator, there will be the structural genes of TRPA to TRPE, there will be the terminator sequence that is marked as small t and t prime, and there is a separate TRPL or TRP leader sequence. There is also a regulatory domain. There is also a regulatory part too that will come into the next slide. Thereby, in the whole structure, the regulatory domain contains the promoter, operator, and the tryptophan leader peptide, or leader sequence rather. And in the structural domain, where from the mRNA will be biosynthesized and followed by the formation of enzymes, the tryptophan biosynthesizing enzymes are fall under the structural domain. Now, what about the TRPL, that is the tryptophan leader sequence? Let us have a look. Within tryptophan leader sequence, there are some regions have been blocked, that is these 4 red rectangular blocks marked as region 1, region 2, region 3 and region 4. In these blocks and the respective nucleotides are region 1 spans from 60 to 68 nucleotide, region 2 75 to 83, similarly region 3 110 to 121, and region 4 126 to 134 nucleotides. Apart from that, just ahead of region 1 there are 2 UGG codons, that is the codons of tryptophan amino acid, and after the region 1 there is UGA, that is a stop codon. Now the point is region 2 can base pair with either region 1 or region 3. So it has the complementary base sequences of region 1 and region 3. Similarly, region 3 can base pair either with region 2 or region 4. Thereby, region 3 has the complementary sequences of region 2 or region 4. Now, how this tryptophan leader sequence will be important in regulating the TRP operon is our point of discussion. Now, TRP operon is regulated at two levels, repression and attenuation. Repression is the first level of regulation. When enough tryptophan amino acid is present in the cell, the operon is repressed. At that time, when tryptophan is available, the repressor protein will combine with the co-repressor, and the repressor co-repressor complex will bind to the operator site. Thereby, the progress of RNA polymerase will be blocked. That is why there will be no gene expression. That is, the biosynthetic enzymes, the enzymes taking part in the tryptophan biosynthesis will not be formed, and no excess amount of tryptophans will be biosynthesized. So that is a very good economical use. Now coming to the second level, that is called attenuation. So attenuation means an immature stoppage. So the TRPL or TRP leader sequence controls this phenomenon of attenuation, and thus is also referred as attenuator sequence. Now what happens? Let's have a look. This is the part of DNA of E. coli with the regions of tryptophan leader sequence, and from the promoter region, RNA polymerase starts transcribing the region, and there is also the UGG and UGA just ahead and after the region 1. So with transcription proceed the formation of mRNA, and as we know, in prokaryotes the transcription and translation are simultaneous events. Ribosome will start synthesizing polypeptide chain on the growing mRNA. Now the ribosome will stall just at the UGA, that is the stop codon, and thus it will occupy the region 2. Thereby the region 3 and region 4 on the hanging mRNA will be free, and as region 3 and region 4 have the complementary base sequences, they will base pair to form a hairpin loop structure. Just remember the hairpin loop structure that has been formed in case of intrinsic termination of transcription that we have studied during study of prokaryotic transcription. So in NSET, this is the image of the representative image of a hairpin loop that is formed by combination of region 3 and region 4. Now this hairpin loop will cause a conformational change in RNA polymerase leading to premature transcription termination, that is attenuation. Therefore, when enough tryptophan amino acid is present in the cell, the transcription of tryptophan structural genes will be attenuated or prematurely stopped. Thereby, no excessive tryptophan will be biosynthesized. Now, if the tryptophan level drops in the cell, then attenuation overcome will take place. What happens? The same tryptophan leader region and the RNA polymerase is ready to transcribe from the promoter region, and there is UGG and UGA just ahead and after of the region 1. Now transcription proceeds, formation of mRNA takes place, and ribosome starts translation. Now ribosome will stop just at the UGG. As tryptophan is absent, there will be no tRNA molecule charged with tryptophan amino acids. Therefore, ribosome will stall just at the UGG codons or tryptophan codons. Thereby, region 1 is occupied by ribosome. Now, region 2 and region 3 are free on the mRNA. As the region 2 and region 3 have complementary base sequences, now region 2 and region 3 will base pair with each other to form the same hairpin loop structure. Now this hairpin loop is further away from the RNA polymerase, that is moving towards the TRPE and other structural genes of tryptophan operon, and this hairpin loop will not have any conformational change, will not have any adverse effect on the progress of RNA polymerase. Thereby the whole mRNA will be biosynthesized, there will be no attenuation, and as the whole mRNA is biosynthesized, the tryptophan biosynthetic enzymes will be translated and the tryptophan biosynthesis will start within the cell. So this is how the level of tryptophan amino acid within cell controls the expression of tryptophan biosynthetic enzymes and thereby tryptophan biosynthesis within E. coli. Thank you.